I would like to pass the floor over to our guest spotlight speaker, Panna Sharma, who is CEO of Lantern Pharma. Lantern Pharma is a clinical stage oncology pharma company developing 3 drug candidates that leverage AI and machine learning to gain a deeper understanding of how to personalize and develop targeted cancer therapies. Lantern in-licenses and develops therapies using genomic data, machine learning in pharma, and computational biology modeling to identify the patient groups most likely to respond to a therapy and to clarify the potential underlying mechanisms of action. So without further ado, I would like to pass the floor over to Panna to give us a brief overview of his company's activities in the AI and drug discovery sector.
Well, thank you, Franco. First of all, I'd like to thank the Deep Knowledge team for inviting me to share what we're doing. I think there's a lot of useful data that they have in their system. It's great to see some of the visualizations. I would love to thank also Dimitri and Andre, especially for recognizing how biotech and pharma are rapidly using AI and data-driven approaches to kind of fundamentally, I think, change the trajectory of drug development. Lantern Pharma is a publicly traded company, so I may make forward-looking statements. We're traded on the NASDAQ as ticker LTRN. And as Dimitri pointed out earlier, we were fortunate enough to go public earlier this year, about 6 months ago. The core of our business, as was mentioned earlier by Franco, is that we look at drugs, especially ones that have been abandoned or failed because there's a lack of knowledge about what patients it'll work best in, or trials do not meet their efficacy or endpoints. And we use big data approaches by assembling all the genomic clinical response data from historical trials, from historical studies, from publications, and also from our own work that we do on those compounds or drug classes to do 2 things: fill in and understand the mechanism of action, understand combinations that could be used, and what patient subgroups and will best respond to that drug. So we use the radar engine now both to stratify patients into subgroups, but also to help us reduce the risk in development. On this presentation, I'll focus more on our AI platform and radar as opposed to the biology and mechanisms of action of our drugs. But I'll try to do it in a way that we accomplish both. When I got to the company a little over 2.5 years ago, radar was about 10, 20 million data points. Today, it's over 1.1 billion data points. It'll probably close the year even north of that. Next year, we expect to get to 3 to 5 billion data points, maybe even more. And our goal is to get to 6 to 10 billion in 2022. And again, that might happen even faster. We're already about 6 months ahead of our schedule. We've highlighted some of the curated data sources. So in addition to bringing in the data, it actually gets cleaned, meta tagged 28 different ways so that we can analyze it. And then we have a specific ontology that we use because we're purely focused on cancer drug development. So unlike a lot of companies in our space, I believe the more focused your problem can be, the greater likelihood of your successes. So the scientific value our platform enables is identifying new compounds that we can rescue and develop, giving us a more nuanced understanding of responder groups, non-responder groups based on biological networks of biomarkers and genes, and feedback for potential mechanisms that we can exploit to do target-based development activity. And that's a very important point of one of our new drugs, LP-184, where we're using that. And by doing that, we're actually compressing what would take in a lab a year or 2, and we're compressing that down to months. And the patient value this has is more rapid entry into specific patient subgroups, potentially with rare and orphan indication, a way to accelerate the trials through companion diagnostics, and a way to improve patient outcomes, which is ultimately the real driver for this, is to save lives and change the healthcare cost. The way we look and communicate radar is important. So right now, we kind of our key architecture platform is ultimately how much blinded success do the algorithms have in answering the questions that we want. And the success rate is now approaching 90%. It's over 85% now. Using the algorithms blinded to answer questions about subtypes of cancer, response groups, genomic networks that are seen, and actual in vivo, in vitro, and also in vitro systems We have about 56,000 patient records, clinical histories, over 144 different drug-tumor interactions modeled. That includes over 90% of the approved cancer drugs, and as I said, over 1.1 billion data points from cancer data sets, both preclinical and clinical. The vast majority, though, are clinical, about 85%. So where do we point this? We point this now to 4 programs. These programs are worth several $billions. The first one in metastatic prostate cancer, that's castration resistant or androgen deprivation resistant. We have our first drug. It's in phase 2 trials today, where it's enrolling. We have LP300, again, another drug that is a very interesting small molecule that's now about to launch in phase 2 for non-small cell lung cancer in never smokers. And again, we use radar to understand the mechanism of action, develop a companion signature, and validate the fact that the proteins that we're targeting are in fact the proteins of interest in the never smoker group. And then a new drug, LP184, which was actually helped with radar from very, very early stages. And we're collaborating with Georgetown and Fox Chase. We have another major collaboration that we'll be announcing. And the same drug, the radar predicted that it would work in glioblastoma as one of the top cancers. And actually it also works in GBM. We went to the wet lab. All this is surrounded by about 106 current patents that are issued and 8 pending applications. So digging into radar, so we bring all this data together from open source, our own proprietary data, private partner data. We cleanse and curate and normalize it. This is very important. Establishing the ontology of what you are looking at helps you understand what kind of questions you can solve. And then we do various algorithms to actually look at the genomic and biological features that we select and that we highlight for analysis, including transcriptome features, DNA features, protein features, features around response, features around blood-brain permeability, immunogenicity. So there's a whole range of features that we look at. And then we, once we understand which ones we're looking at, whether they're preclinical or clinical, we then unleash various algorithms. They can be from XGBoost to artificial neural networks, to scalar vector machines, to simple Keras regression. All these then compete because no one algorithm is perfect. It depends on the question you're trying to solve, the kind of data you have, the timelines and constraints, and these algorithms compete. And then we look at the algorithm and say 2 things. Is it statistically significant, and does it give us biological relevance that will help us in the trials? And then once we have that, that biology and statistical driven feature selection repeats and iterates, and then we finally have really a trained model. That trained model we continue to refine in various other tumor indications, and then we also take that and then apply it to an actual stratification tool or companion diagnostic or biomarker panel that we can use in the trials. Let's look at some of the features of radar. If you look at radar, it predicted, it created a signature. And as the number of biomarkers increased, you can see that the prediction was around 10 biomarkers or so was ideal. And also, as you look at, we looked at the number of different cell lines, and it predicted cell lines exquisitely. The area under the curve for this particular run was pretty amazing. And of course, it really predicted the responders and non-responders in this particular data set perfectly. At this point, we knew we had an algorithm that could be quite compelling. We looked at this algorithm and said, what are the drivers for this 10-gene algorithm? And it really was one major driver, in this case, PTGR1, which is a known enzyme that is involved in very important pathways. And we said, wow, this is great. PTGR1 is, in terms of relative importance, followed by PTPN14 and ASPH. And these were biologically relevant in the tumors that we were looking at. We then went and said, let's look at cancers that have overexpression of PTGR1 to see if this does hold true. Went to the lab, we looked at a number of spheroids and cell lines, and this is across the case. Our drug was several times, thousands or hundreds of times more potent than other drugs. And again, these were all cancers that were overexpressors of PTGR1. When we took PTGR1 and some of those other key genes out, the response rates totally changed. So we knew A, it was dose dependent, it was gene dependent, and that it validated our AI platform data. The AI also suggested that it would be useful in GBM. And again, we went back to the AI to look at whether blood-brain permeability would be a key feature of this drug, and it was. And again, this is a drug that basically, when we went public, we were just beginning to really do this work. And now by the end of the year, 6 months later, we have several programs launched, several collaborations. So that 6 months of work would have taken, I think, in a traditional pharma company about 2 to 3 years and several $1 million more. We then again looked at the PTGR1 transcript levels across a number of solid tumors. And again, we saw that the gene expression, this is a log2 value, correlated with LP184 activity really well. And so this gave us a lot of comfort that this really should be the driver. We did the same thing with another drug. We took, and this is the non-smokers and adenocarcinoma, where we looked at, this is a complex, much more complex mechanism. And we said, well, what really is driving this? And we knew that there was things involved in the inflammatory pathway, things involved in glutathione metabolism, which is really the redox cycle, and then also the ROS pathway. And so as we looked at these, the signature was amazingly mapped to actually these three biologically relevant mechanisms for that drug. And it gave us a lot of comfort that the response signature being developed with 300 was in fact mimicking the biology. And now this drug is now getting ready to launch into a phase 2 in non-small cell lung cancer for never smokers, which is about 20% of the new cases and several billion-dollar treatment opportunity globally. Again, going into the future, as we get more data, it's very important, but data is widely available. So it's really important how the data is put together, how it's cleaned and curated, what kind of questions the data is set up to answer properly. And more importantly, how can you find combinations? Because ultimately, that will be one of the most important ways that cancer is managed in the future. So again, I want to thank you guys at Deep Knowledge. This is just a quick snapshot of what we do at Lantern. Lantern's got some great, amazing, talented people, half computational biologists and AI engineers, and half cancer genome scientists and molecular biologists. And that really is the future of drug development, bringing these two disciplines together rapidly. And so from that, I'd love for actually Deep Knowledge to update our profile in their system, because we have a much more advanced system today than probably their original profile. So we answer a lot of complicated questions, both in preclinical development, clinical trials, and also in patient identification. So with that, thank you for giving me a chance to showcase what we're doing.
Thank you so much, Panna. It's our pleasure entirely. And if you've already registered a profile at the Deep Pharma Intelligence dashboard, you will be able to direct access and submit new data for your company, as everybody who actually registers to this platform will be able to do.